Okay, hi everybody. I want to welcome you to a very special pediatric grand rounds today because this is the annual Mel Grombach uh, Award lecture. And to introduce the Mel Grombach Award and our speaker, I'm going to turn it over to Steve Gittleman.
Thank you, Rafi. Great to see everyone. This is the fullest this room has been in quite a while. So it's great to have this day of show and tell and see what everyone's doing. Very exciting time. So I'm going to start by introducing you to Melvin Grumbach. And, he was here in previous academic generations. So many of you might not know Mel, but he was one of my mentors. And I think he really set the stage for what the department is today. So Mel was a lot of things. He himself was a superstar. He was a great scientist, well noted for some of his seminal observations. But he was also a great coach. And so by coach, he really helped build the pediatric endocrine division, but also the department, and was really instrumental in the institution. So here's a summary of some of his awards. You might not recognize some of these awards. These are the top awards in both pediatrics and endocrinology. This is a small sampling of a very large list. And in terms of leadership, he did some amazing things, including chairman of this department for 20 years. And on the bottom of this, you'll note that he was emeritus. He didn't just come in for a couple of years after his retirement. He was here very active for 22 years, looking up recent articles, making sure the rest of us were up to date. So he was really quite amazing. And I think one of his legacies was training the next generation. And so of the fellows that came through our division, you can see where they went. Again, quite amazing legacy. So this award that we're celebrating today is in his honor. We have a committee that I'm very happy to work with every year, and we try and review the accomplishments of the fellows and figure out who should be the single person to receive an award. I can tell you it's really difficult because so many people are doing such great work, but that's our charge. Here are some of the past winners. You probably can't read this list, but this is a real who's who of pediatric physician scientists. And here's more recent 10 plus years. I think one thing that's really nice to see here is we retain a lot of these people. So if you look around the room, you'll see a lot of Grumbach Award winners who are continuing to do great work. So here are the nominees that I wanted to make sure were recognized for their contributions. I know that they're all giving talks this afternoon, and I'm sure there'll be great talks based on reviewing their Grumbach applications. So Dr. Chapakar, Chu, Higgins, Rainey, and Sitharaman. But unfortunately, we couldn't give an award to everyone, so we had to select one. And this year, the award goes to Aaron Bonansky in critical care. So let me just give a very brief introduction to Aaron and then get him up here to tell his story. So Aaron received his undergraduate degree from Washington University. He then went to medical school at St. Louis University and pursued pediatric residency, first at Texas Children's, and then for his senior year at Children's Hospital Philadelphia. Fortunately for us, he then pursued a critical care fellowship here at UCSF. And he developed a scientific interest, and it became a real personal passion for him to better understand children with multisystem inflammatory syndrome, MISC. And he happened to be in the right time and the right place when this came to the fore during the COVID pandemic. And so, I think it's interesting to see what types of questions people ask and whether they can even answer the questions And Aaron's really done an amazing job in trying to solve this puzzle for all of us. So without further ado, I'll bring Aaron up and let him tell his story.
And first of all, I just want to thank everybody. This is really an incredible honor, and I couldn't be more grateful to have this opportunity to speak to you all today. I'll say one thing, which is when I was listening to these talks this morning and hearing about all of the incredible journeys that the physician scientists earlier were sharing, I reflected on my journey a bit. And while I won't talk in detail about any of it, what I will say is it's been a long, winding road with some bumps in the way. And I just couldn't be more grateful that I'm here and that I found just incredible mentors in both the pediatrics department and then my scientific mentors who are here as well. And I'm really, really grateful to have them. We'll start off with a land acknowledgement, just making sure that we reflect and acknowledge the original people that lived here. And then I'll be talking to you mostly about work I've done trying to better understand the mechanisms which lead to multisystem inflammatory syndrome, but I'm going to try to highlight why you should care about it and why I think it's important, maybe beyond just MISC, which I know there's some thoughts that it may no longer even be a disease that's present anymore. So I'm going to start with just the key players in this talk and try to keep my immunology background relatively brief. But there's two parts of the immune system that are really critical to understanding my work. And the first are antibodies. And antibodies to me are absolutely fascinating. Another highlight that people were talking about earlier today was this idea that you have to really love what you're doing. And I will say that one thing I love about immunology is that the more and more I learn about it and study it, the more I love it. And antibodies to me are a classic example of this. They have the ability to attach to bind to almost anything, which is a really extraordinary ability if you think about the fact that we have a limited number of genes in our body. And so if you think about other ways that the immune system interacts with itself, we think of cytokines. And so if two cells want to interact in the immune system, they use cytokines. And cytokines are genetically encoded. We have roughly 20,000 genes in the human body, and a few hundred of them code for proteins that are called cytokines, and they allow different immune cells to interact. But in contrast, with antibodies, they've found really clever ways to get past being regulated solely by their genetic encoding. And we actually are able to make billions and billions and billions of different antibodies. People estimate you actually can make up to a quintillion antibodies over the course of their life that are different. So that's a million, but a million of those millions. So again, the diversity of your antibody repertoire is just unbelievable. And I think there's a lot we can learn from it, which I'll talk about today. Then there's this idea that antibodies are meant to attack foreign invaders, or that's how we typically think of them. But in certain cases, they can attack the host, and that's what we'll refer to as an autoantibody for this. It's an antibody that binds to part of the human body or a human protein. And lastly, before I lose you in immunology land, I'm just going to briefly talk about what it takes to make what we'll call a good antibody, an antibody that's capable of high affinity binding to its target. And they start out as B cells or precursors to a B cell in the bone marrow. They develop, and early on they have, I guess I don't. Well, I don't know if I have a pointer. They come out of the bone marrow and they have these things on their surface called B cell receptors that give them their specificity. But in order for them to be what we refer to as high affinity class switched antibodies, they need help from T cells. So this is a really important concept, that for a B cell to become able of turning into a plasma cell and making high affinity antibodies, class switch, so going from IgM to IgG, and then affinity maturation, which is the process of really becoming the professional antibody that we think of as being important in protecting against diseases. They need help from a T cell along the way. And then they can become a plasma cell and they can start to secrete antibodies. And so one of the reasons I came to UCSF is during my last year of fellowship, of residency, I was at the Children's Hospital of Philadelphia, and my mentor there was a guy who studied immune dysregulation, Dr. Ed Behrens. And he actually had started a team at the Children's Hospital of Philadelphia called the DIRT team, the Dysregulated Immune Response Team. And we'd go around, and I worked in his lab, and there'd be DIRT team consults, and we'd see patients that were often in the ICU with various forms of severe immune dysregulation. I think these are patients that a lot of us see across different specialties and are aware of. And we categorize them in different buckets. So we'll talk about HLH, HLH-like, MAS, now MISC. And my mentor happened to be an HLH biologist, but what he wanted to get across to me, and what I really took to heart, is this idea that while we can clinically categorize these diseases, we still don't really understand on a fundamental mechanistic basis exactly what is happening in many of these cases. In some of them we do, but in many of them, we're just kind of taking clinical groups and bucketing them together. And then maybe if we could dig even deeper, which is an extremely challenging thing to do, we might be able to develop more targeted therapies and better understand some of the immune dysregulation we understand. And when I was trying to figure out where to go for fellowship, I was asking, what do you guys in the immune dysregulation world think are some really interesting things going on in the world of immunology? And they actually they told me about this guy, Mark Anderson, who was at UCSF, who's become one of my mentors, who studied this disease called APS1, among other things, but discovered that there's this gene called AIR, or the autoimmune regulator. And what this does is it trains your T cells in the thymus to know what's you and what's not you. So it's kind of a deep concept of who you are, but beyond that, it's also really important for immunology. And the really interesting thing is that as you can imagine, if a patient doesn't have functional autoimmune regulator gene, they'll make a lot of T cells that can be autoreactive, but they also make lots of autoantibodies. And again, that goes back to this idea of what I was talking about earlier, that to make an antibody, you also need help from a T cell. And because these patients have these autoreactive T cells, they then generate a wide range of autoreactive antibodies. And in particular, they are known to make autoantibodies that target cytokines. So this really was a fascinating idea to me, because you now have immune dysregulation leading to much more profound immune dysregulation. So the first immune dysregulation is autoantibodies. But the second one is these autoantibodies aren't just targeting, a cell in the pancreas, they're targeting the molecules that regulate your immune system. So this is a kind of wild catch-22 that really fascinated me. And there's some speculation that these autoantibodies, in certain cases, might not just be there purely to cause harm, but actually might be regulating some diseases. And so if we think about the fact that there's a limited number of cytokines out there, but there's an unlimited amount of antibodies, maybe, and this is work that many are exploring now, maybe some of these autoantibodies are regulating the immune system. And so there's a paper that came out showing that patients with this disease had autoantibodies targeting interferons, which is our cytokine, and that higher levels of that actually prevented the formation of type 1 diabetes. Okay, and so just a little visual, the idea is there can now be autoantibodies that target cytokines to prevent their function. And then COVID comes along, and I started my fellowship, and we saw that what I had been interviewing on or talking about as my interest, which is applying immunology and the study of immune dysregulation into critical illness, I got a lot of feedback on various interviews that this is interesting, but how applicable it is. And all of a sudden, we had this new disease caused by a new virus that was causing quite clearly severe immune dysregulation. And no one knew what was causing it or why. And so I really became inspired to try to better understand this, because when I got here at UCSF, we were seeing a lot of these patients. This is an example of actually a patient with a pretty severe course that one of our neighboring institutions took care of, and they sent us samples that we used as one of our index patients to study this disease. But it was a previously healthy 15-year-old that got a mild SARS-CoV-2 infection. And this was kind of a common story we'd hear in MISC. They'd go home, then they'd get a fever, they maybe had some belly pain, they'd go to the pediatrician. The pediatrician would do the correct thing, which is say you're having some fevers, you're having some belly pain, it's probably a virus, go home, hydrate. But then they'd keep getting worse. They'd show up to the hospital and they'd quickly deteriorate. And so we actually saw a number of these patients in our ICUs early on, and we really couldn't explain what was going on, except we knew that they were severely inflamed. And so you can see here just how elevated the lab markers were in this patient when they first showed up. And so the question I had was, how can we try to figure out the etiology of this enigmatic disease that didn't exist until the last few months? And I'll be talking to you about how we went about doing that. But first, I'm just going to quickly talk about what MISC is as a refresher. And so what MISC is it's got a quite broad case definition. So the current CDC will say, if you've had a SARS-CoV-2 infection in the last 60 days, you have a fever, you're requiring hospitalization, you have evidence of inflammation, and you have multi-organ involvement, and you don't have some other better explanation, then you qualify for having MISC. And what it was for what we were seeing was this severe systemic inflammation that was most often leading to critical illness because either the lungs were getting really sick and inflamed, or more often the heart. And so you'd get myocarditis and cardiogenic shock. And another thing that the CDC has noted over the last few years, and this is directly from their website, is that MISC disproportionately was affecting Black and Hispanic populations. And so if you dig through the literature here, these populations were also disproportionately affected by SARS-CoV-2. But even when you account for differences in SARS-CoV-2 rate, there is still a difference in the disproportionate effect of MISC in Black and Hispanic populations. And since we know these are social constructs of being Black and being Hispanic, it would really imply that there are some really important societal factors that we need to think about in better understanding these diseases. And so we made sure that when we were putting our cohort together, that we had a cohort that was representative of the patients that were actually being affected by this disease. And so you can see the composition of our cohort here. I think there's a lot of really important work to do going forward. Okay, so at the very beginning, we were trying to figure out where to go to try to understand this novel enigmatic disease. And one of the common tools for understanding a rare disease is to think about, does it have a genetic component? And so a genomic approach. And it was found in 2020 that about 1% of adults that got critically ill with COVID, and so we were using this to gather some evidence, had these deficiencies in type 1 interferon related genes. And so the idea is type 1 interferons are very important for a viral defense. And so if you have a deficiency that's genetically encoded, then you'd be more at risk of developing severe COVID. But the really fascinating thing was that many more than 1%, here we say 10%, but it's at least 10%, maybe more, actually had autoantibodies targeting interference. And these are those same autoantibodies I was talking about earlier that had previously been primarily described in these rare monogenic autoimmune diseases. But now we are seeing that actually many of the individuals who are getting critically ill from SARS-CoV-2 infections and developing ARDS actually had these same autoantibodies. And the reason this was so interesting to me was that these weren't patients with known autoimmune diseases that had these. These were patients that were seemingly completely healthy, but were walking around the community, likely with these autoantibodies predisposing them to getting this severe disease. And so the first question I wanted to ask was, could these autoantibodies explain this disease? And so the first thing we did was put together a really large cohort of patients that had both critical illness from acute SARS-CoV-2 infection, as well as MIS-C, as well as controls. And to do this, we set up a collaboration with both the CDC and a large consortium of pediatric intensive care units that were being run through this group called the Overcoming COVID-19 Network, which is Adrian Randolph at Harvard was overseeing. And so we reached out and we agreed that let's start by taking a look at all these samples and seeing if these children have this one risk factor. And it turned out that they don't. There was only one patient out of several hundred, 168 kids that were critically ill from SARS-CoV-2, which I'll say is maybe even more rare than MIS-C. Children very, very rarely got critically ill from SARS-CoV-2. And 199 MIS-C patients, and only one of them had this autoantibody. And then we actually went back and it turned out that they had an unappreciated primary immune deficiency. And so this could not explain why these children were getting so sick, and certainly didn't help us understand MIS-C. And so the question became, could there be other autoantibodies to blame? And to answer this question required some new tools and technologies. And so I was talking with my mentor, Dr. Mark Anderson, who we had discussed these type 1 interferon antibodies, and he shared with me there's actually this new tool that we've been validating called FIPSeq in the Derisi lab. And so I thought that sounds really interesting. What does it do? He said, it allows you to look for all autoantibodies at the same time. And I said, that sounds like a great idea. Let's do that. And so the way this technology works is that you're able to take these bacteriophage and you genetically encode them so that they express on the surface a very small piece of a protein. And it can be any protein you want to put into it. But for this project, we primarily focused on a library that the Derisi and Anderson labs had been working on building and validating, which presents human proteins. And so if on each of these bacteriophage, you can present a small piece of a protein, then in our library of around 750,000 different unique bacteriophage, we present little pieces with 25 amino acid overlaps such that they collectively tile the entire human proteome, as well as all known isoforms as of 2016. And so the idea here is you now have in solution little pieces of the entire human protein floating around, which means if you then mix in samples from human serum, if they have an autoantibody, an antibody that will bind to a human protein, it'll stick to that specific bacteriophage that's presenting the protein. And then you can do, you can pull those out do next generation sequencing, and you should be able to see what autoantibodies or what autoantigens are being targeted in these patients. And so we wanted to see, could this help us better understand this disease? And so if we go back to that index patient I talked about at the beginning, he was very, very sick, was on ECMO, but actually recovered, went home, and we looked at their autoantibody profile, and it turned out had extremely high levels of a novel autoantibody we'd never seen before, which is an antibody targeting a protein called SNX8. And so here you can see relative to our 45 controls, and these controls, I'll clarify, are quite important controls because what they are is they're children who got SARS-CoV-2 infection, had a mild infection, and then miraculously, their parents decided to bring them back to get their blood drawn 4 to 6 weeks later, which is really quite a Herculean task to obtain 45 of these samples. But that's the biologic control we wanted for MISC, because these patients have been exposed to SARS-CoV-2 similarly to the MISC patients, and we can now profile their humoral immune response and see if there's differences. And what we found is essentially, well, none of the controls have this autoantibody, and this patient had sky-high autoantibody. And so we thought, this is interesting. Could that potentially contribute? So what is SNX8? And so looked into the literature, and we saw that SNX8 actually is a protein that's involved in an antiviral defense pathway. So that was interesting. But also at around the same time that we were identifying these autoantibodies, a paper came out in which they noticed that a small subset of MISC, the authors believed could be explained by a genetic deficiency, which was to a protein that also regulates the same antiviral pathway that SNX8 is part of. And so this was kind of starting to smell right. There are some interesting features, but it didn't fully make sense because an autoantibody shouldn't be, or we wouldn't predict would be getting inside of a cell and disrupting an intracellular pathway. But we thought this was pretty neat. This is interesting. Let's keep looking. So the next question was, do other patients have this autoantibody? And turns out a whole lot of them do. And so we looked at all 199 patients with MISC. And on the x-axis here, you can see a cartoon of the SNX8 protein. And then on the y-axis, you can see the controls at the top and our 199 MIC patients. And I'm showing reactivity to different parts of the protein. And what you'll see here, I don't know why there's this white thing. What you'll see is about a third of the patients or more have this exact, are targeting this exact same part of SNX8 and none of the controls. And so we thought, okay, we found this differential reactivity in these patients to a human protein that's involved in RNA viral defense. But what about their response to SARS-CoV-2? And this at first didn't seem like a particularly novel question because others had shown that patients with MISC had similar titers of anti-spike antibodies and anti-nucleocapsid protein antibodies. So it seemed like maybe this was a question that had already been answered. But what we could do that was a little different is that the Drisi lab had already built a phage display library, which instead of displaying little pieces of the whole human protein, displayed little pieces of the entire SARS-CoV-2 proteome. Okay, so it allowed us to actually look in much higher resolution at the antibody responses to SARS-CoV-2. And so when we did that, we noticed something pretty interesting, which is that, again, this is a cartoon of the SARS-CoV-2 proteome. And on the far right is the nucleocapsid protein. And any deflection up is a region in which there's much more antigenicity in the MISC patients compared to the controls. And an area going down is where there's much more antigenicity in the typical host response. And what you'll see is, indeed, both the control patients and the MIC patients do have antibodies to the N protein. However, they're targeting a different spot. And this was really interesting to us. And so we thought, could there be some link between the fact that there's this differential humoral immune response in MISC patients and a differential autoantibody response? And so to answer that question, we wanted to really map these epitopes. And we did something called scanning mutagenesis to do this. And this allowed us to find in high resolution the specific amino acids targeted by the antibodies. And it turned out that almost all of the patients had antibodies targeting the exact same region, which I show here. Then we did the same thing with SNX8. And lo and behold, they are targeting an amino acid sequence that looks virtually identical. And so what this indicates is that not only have we found these different responses, but they're probably connected. In the same patients, they have these antibodies that are binding to this shared epitope motif. And this is this idea that maybe now we can start to explain why these antibodies are occurring. They're first having this different response to the nucleocapsid protein, and then that therefore leads to a cross-reactive antibody that also targets a human protein, making it an autoantibody. But we were still went back to this question of saying we'd really like to better understand the actual mechanism of this disease. And it doesn't feel like antibodies targeting an intracellular protein could be the problem. But maybe T cells could be the problem. And again, T cells and antibodies and B cells have to work together to make antibodies. There's other diseases in which there's combined T cell and antibody autoimmunity of the same protein. And so what we've done for the last several years now was try to, in very precise ways, show that these patients not only had autoantibodies, but actually have CD8 T cells that target cells expressing SNX8. And maybe a way to think about this in a disease context that we think about a little more is in, in type 1 diabetes, for example, there will be many autoantibodies to insulin or things made by pancreatic beta cells, but it's not necessarily those antibodies causing problems. It's actually CD8 T cells targeting the cells that make the insulin causing the problems. And so to do this, what we did is we found ways to sort out just those T cells, those CD8 T cells that were binding to the pieces of the protein that were shared between SNX8 and SARS-CoV-2. And I can talk in much detail in the questions about exactly how we did this. And we individually sorted each of these cells and sequenced their T cell receptors. And once we had done that, we were able to show through analysis of the T cell receptor sequencing data that MISC patients had clonal expansions of certain T cells in their blood and quite high levels of these clonal expansions. But then more importantly, we could then clone these T cell receptors back into another cell line and prove beyond any doubt that they are indeed cross-reactive. And so we were able to show that MISC patients have CD8 T cells cross-reactive between this one part of the N protein and this one part of SNX8. And so what we think is going on here is that, there's initially a SARS-CoV-2 infection. And in a typical host, the kids who don't get MISC, they have an antibody and T cell response to the nucleocapsid protein. But in patients with MISC, they have an antibody and T cell response to a slightly different epitope in the N protein, which I draw here, LQLPQG. Because these are cross-reactive, you can also now think of them as autoreactive. These cells are now capable of targeting self. And so these T cells, we believe, are then targeting cells expressing SNX8. And so what are those cells? Well, those are immune cells of the innate immune system of monocyte and macrophage lineage. And many of these are most prevalent in tissue, so in the heart, in the gut. And you can then imagine that these T cells are engaging your immune cells. and then having an expansion, and that likely could lead to a cytokine storm, destruction of these cells, and many of the symptoms of MISC. And so I think there's a few things to think about here. One is this is to me, this is interesting. It helps us to start to better understand the potential etiology of this previously enigmatic disease. But one thing that comes up is MISC is mostly gone. So what's the point of this? And just one interesting thing, and this is very speculative, so I'll say that ahead of time, is that I actually have a separate project where I study the immune responses in patients with Down syndrome. And there was a critically ill patient in the unit last year with Down syndrome who there was some question about whether or not they had MISC. And there was some question, they were ultimately diagnosed with an HLH-like syndrome. I got their blood, I ran FIP-seq on it, and I compared their antibody profiles. And interestingly, this patient actually had the highest SNX8 autoantibody titer that I've ever seen. And so, again, this is very speculative, and I don't want to say that this patient did or did not have MISC. I can't say that. But what I can say is this patient had this autoantibody at very high levels that we've only ever seen before in MISC, that's not present in a couple hundred other patients with Down syndrome I've looked at, and is not present in hundreds of other patients with a variety of other diseases I've looked at. And so I think maybe it's possible that some of these patients we see with continuing somewhat enigmatic inflammatory states may have similarities to some of this MISC biology that we uncovered. And so ultimately where I want to go with this is that I think that we can use this technology and the approaches that were outlined here to try to better understand a range of post-viral inflammatory diseases. And I think we can also use it to try to better understand some traditional autoimmune diseases. And so it's becoming increasingly evident that many classical autoimmune diseases are linked to preceding viral infections. Multiple sclerosis is linked to EBV. Type 1 diabetes is, some people feel, is linked to enteroviral infections. Lupus, there's certain viruses that people think it may be related to. So this is an approach that we've outlined here that actually could help us get at new mechanisms for understanding traditional autoimmune disease. And then finally, in my field, pediatric critical illness, sepsis and ARDS are states of immune dysregulation, and they're disease entities where we are still really trying to understand in a mechanistic way the details of this immune dysregulation. And I think we should be open to the possibility that autoantibodies could play a role. And for example, in adults with ARDS that had SARS-CoV-2 infections, there's been very good data showing that there were lots of anti-cytokine antibodies present in those patients. And so if you can imagine in a state of sepsis or ARDS, if you then have lots of anti-cytokine antibodies, those could contribute and worsen some of the phenotypes that we see. And so what are some direct ways to apply these technologies? And I'll try to finish up quickly, but we can find new biomarkers. So I didn't show this earlier, but early on, we found that with just 3 autoantibodies, we could very effectively classify MISC. And so it's not just MISC, but it turns out the immune system is much smarter than, I was going to say we are, but definitely than I am. I don't want to insult anyone. But the immune system sees what's happening. It observes everything. And now that we have a tool to profile all of the antibody responses of the immune system to self, we can get insights into the specificity of what's actually happening. And this really is an incredible opportunity for new biomarker discovery. And I'll just, without going into too much detail, say that members of the Derisi lab have used this to create models that predict early predictions for cancer. predict who's going to go into preterm labor. There's all sorts of ways in which this has been shown to be true, and I think it's something that we could really do. And so I hope that a future direction is to start looking at the complete autoantibody signature in many of these diseases of inflammation that affect the children we care for. Another thing is we can find risk factors for severe disease. And so biomarkers are one thing, but what if we find something similar to those type 1 interferons that put people at very serious risk of critical illness? And so if we go back and look at some of the data that I generated, but didn't show yet on those children who got critically ill with SARS-CoV-2 acutely, the hypothesis would be that if they have autoantibodies, those aren't because of the virus, but they were already there. And it turns out that these patients do have a unique antibody profile, but it's quite heterogeneous. And so this is an example of one antibody to a tight junction protein that I found in the respiratory epithelium. I haven't done any experiments to show whether or not this does predispose you. But the other thing is that I think if we were to look specifically at anti-cytokine antibodies, we may be able to find a number of things, even though we know anti-interferon antibodies weren't the one that may predispose children to critical illness. And so there's a really talented MD PhD student in the lab now, in the Derisi lab, named Hannah Court-Bowie, who's putting together some technology that should allow us to have much, much better ability to detect anti-cytokine antibodies and circulating antibodies in high throughput. And so, again, I think this could be a really exciting area for some future work to better identify children that are particularly vulnerable. Finally, I'm running out of a little time here, but I think the biggest question going forward is that how are we going to apply the fact that we actually have this new field, which we refer to as autoreactomics? We can now look at the entire autoantibody repertoire all at once. And this emerging field could give us all sorts of new clues about many, many disease entities, including many of the pediatric diseases we study. And in studying this, we wanted to get a baseline sense of what is an autoreactome. And so to do this, we found a way to look at how similar autoreactomes are across samples. And you can see that in this correlation matrix. And we looked at samples from, this is 7 adults who we had samples from over the course of 5 to 10 years. And what we found is actually, and we've now done this in many, many more adults. So this is a finding that holds true. But it turns out everyone has a unique autoreactome. So each of us in this room have our own autoreactome. And many people ask, well, how can you be sure this technology that these are really autoantibodies and this noise in your system? Well, we've done a lot of experiments to show this, one of which is we found that if you deplete plasma cells in patients, their autoreactome disappears. So if you get rid of the cells that make antibodies, your autoreactome disappears. Otherwise, it actually almost never changes. And so you can see your autoreactome is formed and it remains for 5 to 10 years. And so what is that doing? Like, talk about personalized medicine. This is like a completely unique set of real autoantibodies in each of us. And so ultimately what I'd like to do is determine when and why kids develop unique autoreactomes. Is it linked to preceding infections? Do certain features predict development of disease? Do certain features contribute to disease? And so I'll leave it here, but the idea here is that with longitudinal samples, we may be able to predict things such as who's going to get type 1 diabetes well before it occurs. And then we could actually intervene and prevent the development of type 1 diabetes. So thank you so much. I really do want to acknowledge everyone in the Derisi lab, everyone in the Anderson lab, all of my collaborators, my division. I really want to thank my division chief, Jeff. Thanks, Sandrine. Thank Matt for his help, who's also, yeah, so thank you. Be great. Yes, I have about 20 samples of adolescents that develop post-vaccine myocarditis. I haven't published it. There is, they don't have SNCS8. There is some overlap of a different autoantibody, but I don't think that will fully explain it.
Great talk. For the question that we always have with MIC at ID is when someone gets IVIG, and then we try to do diagnostics that rely on antibodies. How did you differentiate between those children with MIC that had IVIG and the autoantibody?
Well, the important thing was to make sure that the autoantibodies we were focusing on weren't due to IVIG. And so all of the autoantibodies that we found by FIPSeq, we orthogonally validated with a number of other assays. And we made sure that on FIPSeq and each other assay, that the same statistical increase was present in patients that had or had not had IVIG. And so there's a panel in the paper that goes through this in detail, but the signal is, we have about 60 or 70 kids that never got IVIG. And so they also have greatly elevated levels of these autoantibodies, which isn't surprising. The IVIG confounding is due to common autoantibodies that get mixed and pooled. And so a lot of this work that I think has criticized autoantibody detection in IVIG, to me, is not valid for the work I do in many ways, because it's showing that you find, common autoantigens in these samples. But if you're looking for novel autoantigens that are very rare, it's much less likely they'd be in the IVIG. But the main answer is we controlled for that.
I'm wondering if you looked for these autoantibodies intracellularly at all. I know that wasn't the pathway, but in myositis, we found that the antibodies do go intracellularly, and maybe that could happen from the COVID infection and tissue.
Yeah, that's right. In myositis, in lupus as well, autoantibodies are internalized, and those lead to inflammatory changes within the cell. We couldn't find any. Those are different processes because, at least to my knowledge, I've not seen evidence anywhere of autoantibodies getting inside cells and maintaining their function and directly disrupting intracellular machinery. And so that hypothesis was so out there that people were less interested in exploring. So I haven't directly looked at it, but I'm isolating patient-derived antibodies. I'm going to be doing that.
Always. I think the part that I hadn't seen before was kind of characterizing these autoreactive T cells, for which like the autoantibody is more of a biomarker rather than being the driver. So I wondered if you have thought about looking, I'm thinking about like more conventional autoantibody-based diseases that we know of, where the patients really don't respond to antibody-based or B-cell targeted therapies and remain with significant disease. So have you thought about ways of kind of leveraging FIPSeq in your methodologies to characterize some of these patients that might be non-responders to like IVIG, plasmapheresis, now earlier or more immediately?
You're talking about kind of the holy grail of T cell biology right now, which is finding ways to detect autoantigen specific T cells. It's really hard. We do believe FIPSeq allows you to narrow down on the proteins you should go after. But more importantly, the Derisi lab and others are working on new technologies to display in high throughput peptides loaded on MHC molecules, which is what's needed to detect a T cell. And so T cell presentation libraries are a much, much more challenging technological feat, but it's something that we're getting very close to, and we're actively working on what diseases to deploy that in. Type 1 diabetes is a great one.
Aaron, just in terms of treatment for MISC, I know it's usually kind of generalized immunotherapy. Do you think this enables you to do something more targeted in reaching these patients?
It does. And so in patients like were MISC still raging and affecting a lot of children, if we didn't have good treatments that were working, I think targeted destruction of these antigen-specific T cells would be the way to go. And there, once you know the antigen, you can design things to actually target those specific T cell receptors. And so that's why so much of the battle is finding the TCR, peptide MHC specificity. And once you have that, there are things in development and things that have been tried to isolate just those T cell populations.
Saw it on one of your slides, although you didn't actually mention it, but macrophage activation syndrome. You see, it's kind of JA. You've probably seen it in the ICU. There's a lot of resemblance to MISC in terms of the way it presents. It's also poorly understood, may have some viral association. I'm just wondering if you've looked at MAS for any of these antibodies.
I'd really, really like to. Yeah. I don't have those cohorts.
I can put you in touch with. We've got a national North American cohort, multi-center clinical trial network, CARA. Some of our hematology group participates in that network with a large collection of serum, PBMC, clinical data. We can help. If you're interested, I can help you.
I'm extremely interested in that. And the other thing that I'd add is we're working on building a newer, I talked about that SARS-CoV-2 library but I'm working on a way to do that with every human virus. And so we could try to see whether for things where we don't know the virus, you might actually be able to narrow a specific differential viral epitope as well as an auto.
If you want to follow up with me afterwards, I can help connect with you.
Yeah, absolutely. Thank you so much. That sounds great. Really appreciate that. Oh,
there might be a time